So, hello everyone. I'm Oliver. Hi, and welcome to another Saturday Microscopy live stream. Happy that you could join me again. And today I would like to put again some bacteria under the microscope. I would like to also do a little bit of so-called gram staining. It's a staining technique that makes bacteria better visible. And I think it's the first time ever that I'm actually wearing a lab coat today. And the reason is because if I get some of the stain on my clothing, then of course I won't be able to get it out anymore. I've seen that some of you have already joined in, and I hope that everything works. Yes, if you have any questions again or comments, please feel free to post them in the chat. Later on, I'm going to answer all off-topic questions, but maybe if you can make it a little bit easier for me, if you have a specific question, please write down at Microbehunter or at Microbehunter Microscopy, so then it's easier for me to find the comments in the chat. Okay. So yeah, true nerd uniform indeed. This is actually the lab coat from very long time ago. You can see that it's already seen a couple of stains before. It's sometimes there are also a couple of holes in the lab coat because of acid that kind of burnt a hole into it. So it's a good protective uniform here as well. Okay, video is great, sound is great. Thank you very much. Okay, so. What I'm going to do today is a little bit actually, I'm going to tell you a little bit of what I'm going to do. I'm going to do a little bit of gram staining. I'm going to also explain to you a little bit of the theory behind it and why it's important. And then later on, if there will be time for that as well, I'm going to put some, yes, I know it sounds weird, I'm going to put some horse poop under the microscope. Manure from a horse. I will explain you later how I got it and why I'm going to put it under the microscope, because yeah, you're going to see later. Okay, so I've prepared actually also something here. So I'm going to start at the very beginning. I'm going to start at the very beginning that if you watched my stream last week, then you will probably remember that I put some, let's put the instructions here out of the way. Then you're probably going to remember that I put a little bit of urine under the microscope. We were looking at some urine crystals and so on. Now the urine over here is now already a week old, and you can see it doesn't look quite as clear anymore, right? It looks very cloudy. Okay. It looks kind of cloudy a little bit, and this actually means that there is bacterial growth. But by the way, I need to give you some additional info. Do you actually see this weird line over here, this black line? Okay. Yeah, just ignore that. That's from my green screen. Okay, if you see, because I've got a green screen behind me, and this is the border of the green screen. Okay, so don't let this weird black line irritate you. Okay, the green screen is just basically behind my back, and this is kind of the border. The frame. That's the correct term, right? So in case you're wondering, right? But I have to adjust this somehow, maybe to get this a little bit. I don't know how. Maybe I'll move over a little bit. I have to readjust this somehow. You know what I'm going to do? This actually irritates me a little bit. So what I'm going to do is I'm going to just a second. I'll get this removed. Yes, this is better because this does look a little bit weird. Okay, so I think that's better. Okay. So that's the thing. There are already questions. Isn't it dangerous to grow and handle bacteria in urine? I'm careful. Yeah, so these are indeed bacteria that where I don't know what they are, right? But this is actually just like if you spoil food, for example, right? So I am careful with those things. However, I don't have a wound or anything, so it should be fine. And this one also is here it's a stationary culture, so it's not actively growing anymore because it's already too old. So what I'm going to do before I do any of this stuff, I'm going to just put this a small sample of this directly under the microscope, simply to show you of what we have here. And then I'll be showing you a short video of the preparation that I have done before, because I did some centrifuging before as well, and so I don't have to do it now. And this is basically where I'm going to start. I'm going to take a very small sample, of this, and I'm going to put it directly on here. Obviously, you know how to make a little bit more maybe. I mean, you already know how to make a slide. Yeah, this one immediately goes off into my trash can. Yeah, cover glass that goes on top, and those slides will then be either discarded That's why I'm using the cheap slides here. You see, these are disposable ones. Normally, I use more high quality slides, which I wash. But those over here, I don't, usually. Okay, so we go on here. And let's put it directly under the microscope and let's see if we're able to find a couple of bacteria. Okay, a pH paper as well, because I would like to measure then also the pH of the urine. Okay, so yeah, let's put it under the microscope. So this means I'm going to go to the microscope view here. I'm going to turn it up. You see again this weird line here in the corner. I'm going to get this away again. I'm just going to change the frame size a little bit here. Up here, no, that's better. It's gone. I've made such a nice setup here with all of the pictures and everything, and then the frame disturbs. So this is basically what I'm able to see here. It is just a regular bright field. You see that there's lots of gunk and goo in here. Okay. So at low magnification, let's go up a little bit here. Okay. Let's go up. You can already see a little bit of movement. And because it's bright field, it should not surprise you that the contrast is fairly low. Okay. So what I'm going to do now is let's have a look at phase contrast. Maybe we're a little bit more lucky there to actually see indeed bacteria. So all of those little dark lines and dots that you see, these are bacteria mostly. When you looked at the mostly rods, these are long bacteria. Yeah. I only have a 40 times phase contrast objective, so of course it would be nicer to have a larger one right here. I have a microscope, another microscope where I'm able to get also 100 times phase contrast. Yeah. So this is and that's basically what we're able to see here. And what you notice is that the bacteria, these are the small dark lines that you see here in the back. All have the same color, obviously, because this is what the microscope delivers, right? So it is not possible to see any differences in their staining because they all have the same color. So what I'm going to do now is I'm going to, if I want to stain them, I want to show you the so-called the gram stain, because this is a staining technique that allows you to distinguish between so-called gram-positive and gram-negative bacteria. It was developed back in the 19th century by a Danish guy. His name was, surprise, Gram. And he wanted to make bacteria visible in lung tissue. So he put lung of people who had a pneumonia, for example. He put those tissue of the lung under the microscope, and he had a real problem. The problem is that there were so many body cells there that he could not see the bacteria, right? The bacteria were so small and few that they were completely covered up by the body cells of the lung tissue. So he could not see them. So he tried around and he invented this gram stain, which is still used today as a diagnostic, right? If you look very carefully, you actually can see that there is some bacteria actually move a little bit. And if you're kind of wondering why there's this stream downwards, that's because the liquid is kind of spreading and evaporating under the microscope. Yeah. So I just wanted to have a quick look here. So plenty of bacteria, and for this reason, of course, we want to be a little bit careful here. So what I'm going to do is I'm going to show you now basically a video of what I have actually done before today. So to save a little bit of time. And I'm going to narrate this video, of course. It looks like this. So this is the urine sample, which is now approximately a week old. I pipetted approximately 10 milliliters of it into my centrifugation tube. And every time when it's a little bit cloudy, this means actually that there are cells present. And then I was spinning it for about, 5 minutes, right? And this was actually a fairly cheap centrifuge. I think I paid around 60 euros for it. And look, when you take it out, I could actually see that there is this white pellet. And the urine, actually the supernatant at the top is clear again because all of the solid particles, the cells were kind of pulled down. Yeah. So here you see at the bottom, you see this at the tip of this pellet, it's called, right? And that's basically 100% bacteria. So yeah, now the video loops. And what I've done is, of course, I poured away the liquid, the supernatant as it's called. And where is now my pellet? Here it is. And this is where we're going to continue. Here it is. This is where we're going to continue now the technique. And so you basically can see I've not used this specimen, these bacteria, to do a gram stain yet. So this is actually going to be the first time that I'm going to be doing this here, right? I mean, I did gram stains before, but I did not prepare a gram stain. Yeah. So the first thing that I have to do here is I have to resuspend this pellet in liquid, okay, in water, and then I have to make a slide. I have to dry the slide, and during the drying time, I'm going to tell you a little bit about about gram staining, a little bit more about the background here. Okay, so during the waiting time, I'll be also reading then some of the comments that you have. Yeah? So I'll be adding a little bit of water now. So, and I want to get everything away here, and here I've got some water, and I need again a tip for my pipette. So here. Go in here like this, and you have now a fresh tip. And then over here next to my computer monitor, I have a trash can where I dispose all of this. In one of my recent videos that I made, people were kind of worried about the plastic waste in agriculture. And yeah, of course, but there's also plenty of plastic waste in the lab. So I'll be adding, oh, that's too little. Look, I'll go up a little bit with the, yeah, let's go up all the way to 50 microliters. Okay, because it's the maximum it can go. And here we go. And I'm just going to be resuspending this whole pellet here by kind of drawing everything, yeah. And yeah, here we go. I see that this is way too dense. This is way too dense. So I have to add significantly more water. But the problem, what I'm going to do is I cannot take this tip now and go back into the water because it's contaminated. So I'm going to dispose it. And I'm going to grab myself a new tip. Yeah, I'll be adding a little bit more without touching it. Okay, here we go. I think it might be better. Okay. So, and I still think that this might be too much. Honestly, it's just from estimation. This is still too much. So what I'm going to do is I'm going to, yeah, I'm going to add yet more. Because if it's too dense, we might have a problem actually. Seeing the individual cells if they're overlapping too much. Okay. This is, I think, better. So, simply, yeah. So, and what I'm doing now is I'm going to put a drop of this bacterial suspension on a microscope slide. Okay. I'm going to put it over here into my rack. And what I do now is the following. Over here, I put a drop on the microscope slide. And I'm going to actually prepare another microscope slide as well. I see that the reflection of the lamp here, of the camera, is not so nice. I'm going to turn the camera off. I think this might be better. Okay. And this one over here, I'm just going to spread a little bit more. Simply to distribute the cells a little bit more. It also speeds up the drying process. And what we're now going to do is we're going to air dry it. And we're going to basically wait until it's dry, okay? And I mean, I do have a warm plate over here to speed this up a little bit. We can do that, I think. Yeah. So I see that I put one over here. It shouldn't get too hot. I think it's too hot. It should really not boil or anything like that. I'm just going to put it on here. And yeah, we have to wait until it's completely dry. And in the meantime, I'm going to have a short look at your comments, if there are any. Okay, so of course there was a hello all around the world at the beginning, yeah. Um, and the question uh, is there a particular reason you don't use the flat-bottomed centrifuge tubes? The point is the following. I'm using, where are they? Well, I've got an empty one over here. I use those tubes here because they actually go into the centrifuge, and the centrifuge is rounded at the bottom, so you cannot stand them up. But I have a rack where I can put them in. Okay. How old is the urine? It's a week old. So I basically collected it last week to make last week's live stream, and then I simply allowed it to stand on my table, and then it started to actually smell like ammonia, because now it doesn't smell so nice. At the beginning, last week it smelled kind of neutral, right? But then the bacteria starts to produce ammonia. Okay, and let me quickly check. Just a second. Here we go. I want to just have to rearrange my windows a little bit here. Okay, just a second. Where is this? Okay, just a second. I need to. Here we go. Okay, so let me see. Yeah, so far there are no. Okay, could you tell us what color band is on what power objective? Yeah, I can also tell you that if you want to. Well, it's now getting finished a little bit, but I'll do that later as well. But I can show you over here in the scope view. If you can, let's go through the whole thing here. Yeah, the red one here, that's 4 times. Okay, so 4 times. Then I've got 10 times, 20 times. 40 times is blue, and then 60 times is also blue, and the next one would be 100 times. It's the white one, but which is the oil immersion objective, which I rarely use. Yeah, but we can actually have a look here now, also at 60 times. Let's try it with DIC. These are again the bacteria. Yeah, here they are. Here they are again. Okay, it's now. Change the colors around a little bit as well. Let me see if I can do it like this. You can see that there is a possibility to adjust the contrast. Yeah. So So it is now basically air dried. So let's move on with the next step. I need to do some heat fixing. So I have over here a bacterial suspension. And the problem with that bacterial suspension is I don't know if it is too dense or not dense enough. I really don't know. That is, maybe I should have checked this before with a microscope. But what I now need to do is I need to heat fix it. And this means is I have to bake the cells, the bacteria, to the glass slide, because otherwise they're going to be washed off during the staining. And normally you do that with a gas burner. I don't have that. I just use an alcohol burner over here. And what I'll be doing is I'll light it up, and then I'll be pulling the slide through the flame a few times, and then I'll be holding it here just to see that it's hot enough, but not too hot. The general rule is it should be just hot enough that you're just barely able to hold it in your hand without actually burning you. So it's pretty hot. Right, but it's not allowed to scorch you, because otherwise the cells are also going to be burned, and you don't want that. What you want to do is by heat fixing it's essentially what you're doing is the proteins in the cells, they will cross link with the glass slide, and the cells will stick to the glass slide. It's a little bit like a meat or an egg in a frying pan. When you heat it up, it's also going to stick to the frying pan, and that's basically what we want to do here as well. Yeah. This is now something where I need and now I have to be careful because up here I've got my camera, so I have to get this a little bit out of the way so that the camera does not get damaged by Okay. Actually, you could also use it just using a gas lighter like this. Yeah. I've also tried that, but I get a little bit more control. Yeah, this way over here. So I'm going to carefully heat it up. Test it. Not yet. Yes, this should be okay. Okay. So I now need to have a place to cool it off. I'm just going to put it on here over on the water here. Okay. I just realized that I forgot to actually tell you the whole process. So I'm finished. Yeah, so this is yeah, basically heat fixed now. Okay, and I hope that the cells are now all properly cross-linked to the glass slide. So I just want to show you now the steps, because I actually did prepare a little, how do you say, a little sheet here. Yeah, so heat fixing, so that's the first thing. And now I need to add crystal violet, which is the primary stain. It's a pretty strong stain. And in order not to get any stain on here on my table, I'm going to take a little bit of tissue paper and put that down on here. Okay. I see that there are a couple of questions about connecting cameras to microscopes. I did make a couple of videos on those. DSLR camera to a microscope is a little bit of a science in itself. There are a couple of possibilities. Not all solutions work equally well. Okay, so this is now step one, crystal violet. Okay, this is you already know the stain because I've been using it in the past, and it's a pretty powerful stain. And what I want to normally do now is I'm going to simply use a pipette. Well, or yeah, you can use a pipette if you want to, and you can just basically carefully spread it. That's going to be enough over here as well. Again, I have to tell you, those live streams, when I do them, sometimes it works well, sometimes it doesn't. That's always a little bit of a, how do you say, there are always a couple of unexpected elements as well. So we're going to see how well this actually works, right? So I usually give it a minute or so for staining. Okay. Yeah. So I'll just check a little bit. Yeah. Yeah. Okay. So the modern Victorian. Okay. Yes. I guess this response. I will find that one out later. I guess you're referring to the videos that I made with connecting. I think I put some of them actually into a separate playlist as well. Yeah. Yeah. So that is basically. You see, it already also starts to dry a little bit. And now the next step is also quite interesting. And the next step is I have to add iodine to it. And what it will do now is the following: the iodine will react with the crystal violet, and it will form a so-called a complex. Yeah. So this means the crystal violet has entered the cells, and the iodine that I add will now react with the crystal violet, and it will basically make sure that the crystal violet will stay inside the cell. Maybe, okay, because that's going to be the gram reaction. Yeah, because if it stays inside the cell, then it's a gram positive. And here is my iodine. And you got to be careful with the iodine. This is pretty aggressive stuff, and it's interesting. And this was actually rubber, but I cannot push it together anymore because it became so brittle and so hard over the years that I think the iodine somehow must have reacted with it. So what I need to do now is I need to carefully open it, and I'm going to use another pipette because I cannot use this anymore. I have to use another pipette to actually get some of the iodine out. That's a lot of iodine. Immediately close the bottle again so that it doesn't fall over. And I'm going to now add the iodine directly to the crystal violet. And there is this kind of reaction happening. Okay. Okay, and I think that should be enough. And the next thing is the following. Okay, so this now reacts. You can actually see that it's almost like it starts to form almost like a skin or a layer on top of it. Yeah. So this is the reaction that's happening. Okay, should be okay. And I really hope that this is going to work. The pipette I'm going to put somewhere where it doesn't disturb. And the next thing is the following, and now I have to do the so-called the washing step right over here, right? Iodine, decolorize with ethanol. So that's very critical because gram-positive bacteria will not be basically not be washed out. Okay, so the stain will stay inside gram-positive bacteria, And inside from gram-negative bacteria, the stain will be removed from the cells, and the cells will lose again the color. So that's this step number 4. Now, how am I going to do that? For that, you need a trash can, a beaker over here, right? Which acts. I'm going to move this up a little bit here, which acts as a as a collection. And then you do the following. You go over here like this, and over here I've got ethanol. It's pure alcohol, and you just carefully let the alcohol run over the glass slide until it is completely decolorized. Okay, so that's really, it might take a little bit of time. Let's move over here as well, because it's you have to do this until no more color is given off. You see, as soon as there's some blue stain runs off, it's not finished yet. Okay. Okay. Maybe allow it to Of course, you don't want to spray it directly, because then you might be washing off the cells as well. It almost becomes completely clear again. It would be kind of embarrassing if the whole thing wouldn't work. That's always one of the, how do you say, the stress factors that I'm experiencing. I want to do a demonstration, and I don't know, then maybe I'm dropping the slide in here, or I don't know, or something doesn't work, or I forgot a step, or, you know, it's lab work is never 100%, yeah. See that there's still some, yeah. Could be enough, maybe. Okay, I'm going to leave it as it is now. Okay, and the other one here as well. That's the critical step. Okay, let's give it a little bit time. Yeah. There's a question over here. What is the function of the iodine in this procedure? The iodine is referred to as a so-called a mordant. Interesting name. What it does, it reacts with the crystal violet, and it forms a so-called a chemical complex inside the cell. So it will retain the crystal violet inside the cell. It will kind of bind it inside the cell. And only a gram-negative bacteria can be washed out again. Yeah, so it kind of, so it kind of makes sure that the crystal violet stays inside the cell. Yeah, and now this step here is now, so to say, the determines now whether the bacteria are gram-positive or negative, because gram-positive bacteria, they will keep the stain, and the gram-negative bacteria will be washed out. Okay, so let's see. I really hope that this works. I did a couple of months ago, I did also a live stream where I did gram staining. And in that live stream, it actually did work quite well. Okay. Of course, I used different bacteria at that time. I need to do another one over here. Okay, let's assume that this works. Okay, so the next step. Okay, so you see that it's still a little bit blue here, right? We can make it a little bit larger. This one over here is still a little bit dark. I don't know if everything's been washed out, but maybe we're just going to leave it as a reference a little bit. And then the next step is the following. Now, those gram-negative bacteria have lost the stain again and cannot be seen anymore, right? That's a little bit the problem, right? And so what I have to do is do the so-called a counterstain. And yeah, that's this step over here, number 5, is the so-called the counterstain. The counterstain makes sure that the gram-negative bacteria are visible. And you're using a red stain called safranin. And yeah, so what I'm going to do now is I'm going to quickly, because this is how do you say it, is still covered with alcohol, it's already evaporated. I'm going to now add a little bit of safranin directly on here. You see that this one over here actually is way darker. You know what, I might have to decolorize it a little bit more. Yeah, there's still some stain coming out. If I don't do that in everything will be blue. Okay, so, but I'm going to leave it as such and hope for the best. And now the counter stain. Okay, and again I will spread it a little bit using a pipette. And so usually again a minute or so should be enough. And then I'm going to rinse everything with water. Okay, so let's have a look here. Yeah. So can you use isopropyl alcohol instead of ethanol? I think both should be possible. I'm always using ethanol because it's simply cheaper. Yeah, and there's been a question here, what's Oliver doing? I'm doing a so-called gram stain. Okay, and this basically is a traditional stain in microbiology. It's a bacterial stain, and I really hope that this works out. Otherwise, it's going to be a little bit embarrassing for me if it doesn't work out. Otherwise, yeah. Yeah, so let's see that the stain spreads properly. That's the counter stain. Yeah, and what we should see is we should see, hopefully, if everything works out, we should see a mix of blue and black and red bacteria. So now this is now water. Okay, this is now water. Okay, number 1 and number 2. Okay. I just realized maybe we don't even need the heat plate. So, and put everything away here. And now you have to air dry it. Okay, so what I'm going to do now is, yeah, and here as well. And the air drying is of course takes a little bit of time. It has to be dry because I want to use oil immersion. So and for this reason there should not be any water on here. So what I'm going to do now is I'm just going to put it on the hot plate here, or on the warm plate. Yeah, and you know what? If I carefully do it like this without, then it should also be okay. Let's put it on here. Let's turn this also carefully around to absorb just the most of the water. So, the staining process is finished, and as I wait for the slides to dry, I'm going to quickly show you a tree of life. Okay, because so that we, so that's basically the biology lesson now. Okay, and by the way, here are the gram positives. So that's the reason why I want to show it to you. So basically, living things can be categorized roughly in 3 different domains: bacteria, archaea, and eukarya. We humans are over here in the animals. You see, we're quite related to the fungi and the plants and so on, right? And over here, you've got the bacteria and the archaea. These are also prokaryotes. They basically live in volcanic hot areas. But the gram positives, you see that they have a separate branch over here, and the others are gram negative. So it's kind of nice because by doing the staining procedure, we can actually say something into which category they belong, right? This is basically then the idea that this allows you a little bit something to tell you in which place among the bacteria those those that we have are, right? Because just by looking at the microscope itself, just at the shape, the shape itself doesn't tell us a lot. But the gram positives are over here, And yeah, so if we see some of them having stained completely black, then they belong to over here. So, and now everything is dry, and now I'm really nervous, and I am really nervous. Okay, I'm not just saying that, because I really hope that this actually works out. So I'm going to remove those here. I'm going to turn off the hot plate again. I mean, you see that there is something on here, so something did stain. And my hope is to be able to see at least blue, black, and red stained bacteria. So let's put this one over here. I don't know. Let's start with this one over here. It's very dark, so I think maybe a little bit too much over here. And now to this view over here. Okay, and yeah, we already see something over here. And let's have a look. Let's zoom in a little bit. Okay. Okay, I think we need to go up a little bit higher. I need to remove my polarization here. So, and I'm going to go 60 times. Okay, and I think I need to use oil immersion. I'm kind of happy already. But we do not see, at least here, at this magnification, I do not see a lot of color difference. So, but let's have a look. You see that some of the bacteria are stained a little bit red, and some of them are stained more darker. You see, especially, maybe that's a good one. Look, if you go out of focus, you're going to see that some bacteria are actually black, right? And others are reddish. And the black ones, these are the gram-positives. Okay, so actually I am kind of happy with the result here. You know what I'm going to do is I have to use now not phase contrast. I have to now use oil immersion. So I'm going to put in my oil immersion objective without oil. I'm focusing, and this is pretty bad, right? We don't see, it's kind of blurry. I now engage the focus lock lever. Okay, so it will remember the position. I lower the stage without cover glass. Without cover glass, I directly apply some immersion oil. Now you have to check what I'm doing up there. Directly on the slide, on the bacteria. Now you know why it has to be completely dry. I raise the stage with the coarse focus. It will remember, it has remembered the top position. And therefore, here we go. Okay. And indeed, I think I'm quite happy with the result. Okay. Actually, I'm very happy and I'm relieved as well. Okay, I'm quite relieved as well because it actually did work quite well. So I'm going to explain what you see here. Okay. Yeah, this is actually quite nice. Okay, good. Very good. But the result doesn't make me happy because I see a lot of gram-negative bacteria here. And they're not always very good. Okay, so let's have a look. You see over here, let me start the arrow. Okay, obviously, you see all those dark things that you see over here. These are darkly stained bacteria. They're the gram-positives, right? And then you see those others that have stained red. These are gram-negative bacteria. Yeah. So a lot of so-called gram-positive rods. The long bacteria are the so-called rods. Yeah. Over here as well, you see, also quite nicely. Yeah, that those bacteria stain differently. Yeah, also here quite nice. You see over here, gram-positives, and between here, those red ones. Oh, here as well, those red ones, all gram-negatives. Yeah. So actually quite a nice result. Okay. So it is, has as expected and as hoped for a nice mix here. I also would like to try it in the other slide, because this one is the slide where I had which was very concentrated. Yeah. But I think as a demonstration, I can say I'm quite happy. Now you probably want to know, okay, but besides gram positives and gram negatives, what are they now? And that's, I tell you, then you have to go really deep into bacterial identification. You have to make pure cultures. You have to grow them in pure cultures. Probably do DNA analysis, and so on and so on and so on. Yeah, yeah. I don't know. Maybe there are a couple of hundred different bacterial species even in here, right? So this is also quite a nice place here. You can see, yeah. Yeah, all of those tiny bacteria over here, yeah, are gram negatives, and those larger ones gram positives. So not a lot of gram positive cocci around cells, mostly gram positive rods. Now, when I did this a long time ago, during my university time still, doing I made lots of gram stains, but if you want to get this to publication level, then you have to grow the bacteria in pure culture. You have to actually do the test at the right time. That's also important, because they stain differently depending on whether the culture is young or old. Oh, here, okay, this is too dense, for example, over here, right? Yeah. So there are plenty of factors that you have to standardize as well. Yeah. Yeah. So yeah, lots of gram negative. So I'm quite satisfied with that. Yeah. Yeah. Here again, more gram positives. Now you have to understand the following, that there, as in so many cases, there's a natural progression of bacteria. So the pH of the urine changes or has changed over the days. So this means that, of course, different bacteria start to grow and others start to disappear. Yeah. So, and I would like to now try the other slide. Okay, so the other slide has a lower bacterial density. So the same thing. I'm going to lower now the thing again here. Drop of immersion oil directly on here. You could not see that because over here I have to center it. And we go up here again, and I hope that I'm able to find the focus. Whoop. Okay, so this is the one where I had less of the sample. Okay, I like the other one better. Oh, okay, here it's better. Okay. Yeah, it's similar to the other one, not quite as dense. Not quite as dense. I think that I have what I could do is I could grow myself some E. coli cells. I've got a safe source of E. coli cells, and they stain gram negative. Yeah, I did like the other one a little bit more. Because yeah, so you see even here there is a little bit of trial and error involved, but if you find maybe a place, over here you can see that there are some cells. I don't know if it also comes across YouTube quite well, but there are some red cells over here as well. Yeah. So what I'm going to do now is I'm going to quickly read some of the comments. Well, actually, Michael, my son, he's actually preparing a comments list again. Okay. I'm just scanning through the whole thing. Yeah. So there is a little bit of a question of which alcohol you can use. Yeah. A safe source of E. coli cells. I should explain this. Yes. Yes. The thing is the following: E. coli and E. coli. Obviously, when you use laboratory grade E. coli, then they should be safe. They are safe. It's like this, that it is possible to buy E. coli in the drugstore, so-called freeze-dried E. coli cells. You can for eating, and people swallow them to regenerate their digestive system after they have been on an antibiotic treatment. So these are safe, freeze-dried E. coli, and I have already grown them at home. So basically, I've opened one of those capsules with those E. coli, and I put it on a inoculation loop, and I made a plate, and they started to grow, right? And that's basically something that I have worked with already before. Because what I do not want to do is I don't want to grow otherwise in a culture medium on a petri dish. I don't want to grow otherwise other bacteria that might be also able to grow on the skin and on the body. So I want to only, if I play around with bacteria, only the safe sources. For example, plenty of bacteria can be found in yogurt, lactobacillus and so on. Of course, a safe source. People make their own yogurt at home, grow their own yogurt bacteria at home. On cheese, for example, certain cheeses, they contain bacteria. So all of the bacteria that are used for food, that's going to be fine. Yeah. Okay. What does some biology info? What does gram-positive and gram-negative actually mean for a species of bacteria? Yes, I wanted to explain this to you as well. So I need a sheet of paper here, and I would like to now tell you a little bit of what this actually means and how this actually looks on a cellular level. So let's go back to the desk view. So let's go here. So this is going to be again a short 5-minute biology lesson, but don't worry. Okay. So when so first of all, the word gram is by Mr. Gram, the Danish biologist who back in the 19th century who discovered this one, and basically it's been used like he invented it. Apparently, I did a little research. He's been experimenting around, and then he discovered this, and it's been used in pretty much the same way ever since. And if the cells, the bacteria, stain blue or black, they're referred to as gram-positive. And if they stain red, they are referred to as gram-negative. And red is because of the safranin. So that makes it red. And the blue-black that makes the crystal violet, makes it blue-black. And then you decolorize it with ethanol. You wash it. And the gram-positives, they will keep the blue-black stain, and the gram-negatives, they will lose the blue-black stain. And why does this happen? Because they have a different structure of a cell wall and cell membrane. So the gram-positives, what they have is the cells have over here a cell membrane, a plasma membrane, and on the outside, they have a very thick cell wall. It's the cell wall. And this in here, that's the cell membrane. Cell membranes. And they will stain gram-positive. Okay? And the reason is the following, is because when the inside here turns blue, what will happen is because of the thick cell wall, the blue color cannot be removed with the alcohol. It will stay blue because the cell wall protects it. Okay? So, and then you have the gram-negatives, and what they have is they actually have two membranes. They have two membranes, and in between here, they have a thin cell wall. Okay, they have a thin, so the inside, that's the cell wall. And here you have an outer membrane, an OM, that's the outer membrane, and the inner membrane. And what happens now is if you add alcohol, then the alcohol is going to destroy the outer membrane. It's going to dissolve it. And the cell wall itself is not thick enough and strong enough to actually keep the blue color. So what's going to happen is the blue color will be washed out because the alcohol removes the outer membrane and it washes out the blue color. And then when you add safranin, it will turn red. Yeah? So this stain, this gram stain actually tells you something about the structure of the cell. And why is this? Does this have any medical importance, you might ask? And very much so, because the gram-positive cells are more antibiotic sensitive. So if you're infected by a gram-negative one, if you've got a gram-negative infection, then the antibiotics are not going to harm it so much because of the two cell membranes that it has. It prevents the antibiotics from entering the cell and killing the cell. Yeah? So antibiotics, certain certain antibiotics, like penicillin, for example, they're able to kill off the gram-positive bacteria much better than the gram-negatives. Yeah. So just wanted to give you a short biology lesson here, and this means that over here the gram-positives, these are the ones, the bacteria that have one cell membrane and a 6-cell wall on the outside. Yeah. So the staining and the way that they stain actually reflects something or tells us something about the way that the cell looks like. Okay. Yeah. Does the ethanol denature the cell protein, preventing the pigment from escaping? First of all, ethanol does denature proteins. However, because it did the heat fixing, the proteins of the cells are already denatured. So, the fact that ethanol denatures the proteins is correct, but that is not the reason why protein denaturing has nothing to do apparently with destaining, but it has to do something with the structure of the cell wall and the cell membranes. Yeah? So, thick peptidoglycan layer. That's correct. That's the cell wall is made of a substance called peptidoglycan. Yeah. So this is just a very short crash course, what I want to do now is, of course, I want to go back a little bit just to enjoy a little bit the how do you say I'm going to do now the following again. I'm going to go back to the other one and then I want to move on. Yeah, a little bit. Here we go. That's the other slide. This one is actually better. The slightly denser one is actually better. Yeah. I'm fairly happy. I'm fairly happy with the result. What actually happens if I were to turn on? No, it's not good. That's not DIC. No. You see, that's DIC, differential interference is not always better. You want to use bright field for this. So this is regular bright field. Yeah. So I think also over here. Yeah, plenty of I see that the gram negatives are actually much smaller over here. You know what I'm going to do now? I'm going to try something. I hope you don't mind. Look, I'm going to try something. Where is this? Here. Just a second. I'm going to try to zoom in. I'm going to try to... Does this work? I don't know if this works. No, it's too small. Can I make this bigger somehow? How can I make this bigger? Let me experiment a little bit. I want to make this a little bit bigger, maybe. I don't know. You see what I'm doing? I'm trying to make it becomes blurrier and blurrier. But maybe. Okay. Yeah. Here we go. Just a second. I'm playing around now with the program. How can I make it larger down here as well? Okay, but I think I think that might be better. Okay, you can now see that the smaller gram negative cells much better as well. That's an interesting one over here. 50 years ago, when they taught us about gram stains, there was a rule of thumb: gram positives are pathogens, gram negatives may not be pathogens. Sorry, that's not anymore the state of the knowledge, current state of knowledge. Whether something is a pathogen or not, it depends on the so-called on factors, certain virulence factors, and not whether they're gram positives or negatives. There can be many dangerous gram positive pathogens and also gram negative pathogens. I'll give you an example. For example, the lactobacillus, which is found in yogurt, is gram positive, and we can eat it, right? It depends not so much on gram positives and negatives, but actually on whether they actually are dangerous or not. Yeah. So So this is the I think you get the idea. I have to tell you personally, I'm very relieved and happy. I did not expect quite such nice results. Yeah. What is the approximate size of bacteria? Also a good question. Rule of thumb, bacteria has 1 micrometer. So that's we're talking about order of magnitudes. So if you take a millimeter, Yeah, you know how big a millimeter is. And if you divide it into 1,000 sections, right, times, you get 1 micrometer. That's approximately the size. And a human cell approximately has 10 micrometers across, has 10 times the diameter. So if this basically were now, let's say, I don't have a scale now, but if this is now, let's say, were 1 micrometer, yeah, 1 micrometer across, then a human cell would be about this size. Okay. Well, I don't know, a blood cell has about 8 to 10 micrometers across. Yeah. So, yeah. So this is the, yeah. So 0.5 micrometers also possible. Yeah, there are some that are significantly smaller, some significantly larger. Yeah. But yeah, in that range. Okay, I'm going to move on. Ooh, it's already 1 hour. Okay, 1 hour gram staining. And I did prepare something else that I want to show you. And we're going to do this very quickly, but it's going to be a very, very, how do you say, big change in topics, if you don't mind. Okay, so I'm going to, yeah, I'm going to make a big change in topics. And I'm going to do this again by showing you a short video. Okay, so I'm gonna, the reason why I decided to show it to you today as well, because I'm working on this as well. And yeah, here is the video. One of my students, they have horses at home. And she wants to do a little investigation. This is horse poop. Okay, this is horse manure. And she wants to do an investigation on checking if there are any parasite eggs in the horse manure. Okay, so she took along a little bit of this horse poop, horse manure, and we in school tried to extract some parasite eggs, and we were kind of successful. And then I said, "It's okay, cool. Now I also have something to show you for the live stream." Because they actually are doing this at home too. Yeah, that's the new sampling one. Because at home they're doing this actually because they have to check whether their horses have parasites or not. And she wants to convert this into a biology experiment. So she brought along some horse manure. I think this is the first time ever that somebody is picking up horse manure with chopsticks. But it turned out to be very practical. Okay. And I also have to say the following. It does not smell bad, really. It smells like stable, like grass and stable, like a farm. It was not disgusting. I just have to clarify this. Horses are vegetarians, so it's purely plant material, and you cannot compare this to the excrements of carnivores, dogs, cats, and so on, because that can actually be dogs, cats. You want to steer clear of those, right? I'm adding now concentrated salt water. And because I want to extract, I want to extract some of the eggs. Normally, there is a strict protocol how much of the feces you have to add with how much liquid. I'm just doing this a little bit for demonstration purposes. Okay, so this is not the ideal way that you should do it, right? And then you have basically dissolve all of this manure, and then you, of course, you put it through a strainer. Thank you very much. I got some questions again from you, right? And yeah, so again I have to say the following: I wouldn't be doing this if it were disgusting. Luckily, it was not. Yeah, yeah. So this is the Klebsiella pneumoniae gives me stubborn urine infections. Klebsiella is gram-negative. Yep. Yes. Yeah. So yeah. And yeah, so basically I washed out whatever is in here. And then essentially what I did as a next step is I filled it into tubes. Now the question is why did I use salt water and why did I use concentrated salt water? And the reason is because I wanted to do a so-called a flotation technique. Yeah. Yeah. What happened after the gram staining of the bacteria? Okay, yeah. So the thing is I basically extracted now whatever was able to be dissolved, and I filled it into one of my centrifugation tubes. Okay, so that's basically what I've done here. Yeah. And then I let it stand for half an hour. Actually, you're supposed to let it stand for half an hour. I let it stand for a couple of hours. Not a good idea maybe, but I've got it here. Okay, so this is basically where where we're now going to continue. So very carefully, without shaking it, yeah, here is the tube. Okay, I'm going to move this over a little bit so that you can see what I've got here. So you can see at the very bottom, very difficult to see, solid material has settled. And in the salt water, because salt water is more dense, yeah, what should have happened is that any parasite eggs that are in here should have started to float to the top, okay? And they should be up here. So what you're supposed to do now is you're supposed to take a sample from the top here and put it under the microscope, hoping to find or hoping not to, depending, yeah, at least I hope to be able to find a few parasite eggs, round structures, okay? These are which parasites are very often nematode worms. Now, the protocol says you should not let it stand too long because the salt water is not good and so on. I did not meet that protocol. I just let it stand a little too long, maybe. Okay, so we're gonna try it in any case. Okay. Yeah. So yeah, why should the parasite eggs float? Important question, because they are less dense than water. Yeah, you can actually, I did not know this as well, you can actually buy if you have horses, you can actually buy those solutions yourself. There's even a special microscope slide with a scale, a grid, that is, so that you can actually also count the parasite eggs, and that's actually the density. Yes, density differentiation. Yeah, and yeah. So what I'm going to do is the following, and I'm going to open it, and it does not smell bad. Honestly, it smells like horse. Really, it's yeah. Again, a horse is only eats is a vegetarian. Yeah, yeah. It was salt water. I used salt water, and apparently you can also use a glucose solution. But I don't know. I really hope that this is actually. I haven't disturbed it too much. And I put it on here. And I'm happy if I'm able to find only a few of them. Yeah, however, I asked my student, and she said she's in some horses she found 50 of them, right? And what her investigation is the following: she wants to determine whether the number of parasite eggs is age dependent, yeah, or horse age dependent. Yeah, I used concentrated salt water, so I simply added salt until no more salt could dissolve. I did not even measure it out. Yeah. So, yeah, interesting. Yeah, thank you for that. We've tested hedgehog feces for roundworm eggs this way. Thank you very much. That's interesting. Okay, so I have to now remove again the microscope, the thing over here. So these are basically I need to lower this, and I'm going to remove the slides, and I have to, of course, change the. How do you say? I have to change the view again. Just a second. I have to just a second. Here we go. Okay, I have to go back to the original size, and here it is. And we're now going to hope for the best again. Okay, I have to find the focus, and it's way too bright. Just a second. Oh yeah, maybe that's the one. Nope. Just a second. I'm going to do it like this. I'm going to do it here. See, I have some issues focusing. Some bubbles. Bubbles are always good because bubbles help you to focus. Okay. So here we go. We need to go up with the magnification. Okay, so bubbles help me focus. We see a lot of debris and lots of stuff, and yeah, you get the idea. And I'm going to now do this here. And what we have to do now is we have to now search for some round structures, and I'm not talking about the air bubbles. Yeah. And if this doesn't work, then I'm going to take another sample where I have found stuff. Okay, so lots of manure. I have even found microplastics in here. I kid you not. It's not surprising, really. Yeah. But if I don't find any over here, then maybe I was shaking a little too much. This is an air bubble. Okay. So look at maybe. Okay. This could be one. I've seen others that look a little bit nicer, but do you see this round thing in the center, right? Yeah. Actually, there's a catalog of different shapes of eggs. Yeah. They can be oval, they can be completely round, and depending on on the shape, you can guesstimate of what they actually are. Yeah. But this one here is not so super dense. I've actually, when I tried this with this one could be an empty one maybe. This round one. Do you see this? Yeah. This one in the center right now. Yeah. And basically, when you have now a grid on the slide, then you can do an estimate on how many parasite eggs there are present. This one looks again like more like an air bubble. Okay. So what I'm going to do now is it's already over an hour. I'm going to now take a sample, which I did not, which I'm basically, where I'm going to simply shake it a little bit, shake it up a little bit, and where I'm not going to use just the top layer, but actually also the layer beneath it. Now see, there are not a lot. I've seen maybe 2 or so, but I mean, you get the idea. Ah, here are the 2. Huh. Here, happy. Yeah. Look. Here's one, and here's another one. Yeah. So there are a few present, and you can see that they look quite different and also larger from the other debris, that we have here. They will, they will unfortunately, I've just read the comments, they will not hatch because I treated them with salt water. However, it actually said in the instructions that you should actually cool and the feces of the horse should be fresh and you should cool it, otherwise they are going to hatch. So it's indeed a thing. Okay, over here looks like another one, right? Very clearly not an air bubble because you can actually see that something in there is in there. Yeah? Yeah? I suspect that this is a species that only lives in horses. Honestly, would not surprise me. I mean, some of the parasites or also even bacterians are so highly adapted to a specific species that you cannot find them anywhere else. Would not surprise me. Yeah. Okay, so very quickly I'm going to do the following. I'm going to now take this sample over here. Yeah, and you see that there is a little bit of stuff that has collected here on the bottom. So what I'm going to do now is, can I actually go all the way in? That's the question, or is it a little too... Yeah, I have to be careful. Okay, from the length it might work. I want to take something of the solid material here on the bottom. It's a very dense material, and we're going to see if we find something in there as well. So you can actually see what I'm doing here. Okay, let's see if there's something in here. So look, the desk view again. So this is what I have here. I don't know if this is very, I don't know. This does not look very... I'm going to try something different. I'm going to try to agitate this a little bit. Maybe there's a little bit more stuff here now. Yes, it looks a little bit more. And let's take this one here just as for demonstration reasons. Here we go, a little too much liquid. So I'm going to remove some of this liquid here. because I don't want the cover glass to completely float around too much. So let's give this here a try. Because I've seen also some interesting plant material even. Oh, yeah. Okay. Okay, it's still floating. Ah, yeah, over here you see one. Yeah, you get the idea. Here's another one. Looks like an air bubble. Okay, over here might be one. Okay. Yeah, so you basically can, you can hunt for parasite eggs this way. Yeah. Here's one. Actually, a lot of them. And it does take a little bit of time to systematically actually count all of them if you want to do some statistics. Yeah. I was hoping for this. Look at this. I love it. What do you see here? I wonder if some of you might, of course, some of you might know what this is here. This weird structure. It's one of my favorite. Almost every live stream I find, not everyone, but very often. Yeah, this is a so-called xylem. It's from a plant tissue which transports the water, and very often in the cell wall they have those spiral-shaped strengthening structures, and they're difficult to digest. And therefore you often find them. So even yesterday and today, I found several of those spiral-shaped xylem structures. Horses, of course, eating plants. Yeah, they digest it, but those spiral-shaped xylem structures is quite strong. Yeah, so you occasionally find those. Yeah. Yeah. So that's cannot believe it. It's we're looking at horse poop here. Yeah. That's an interesting one here as well. Yeah, maybe also again, as I said before, I have also occasionally found some stuff that should not be in there, like an egg in the center here, like some microplastics or plastic fibers. But this one seems to be better. Okay. Yeah. Here, another round one. Another egg. Here's one. So maybe the flotation technique, maybe I did something wrong. That it was not able to concentrate the eggs as much as when you take it from a sample, we just mixed it. Here, do you see another xylem? Yeah, over here. So okay. So what I'm going to do now is I'm going to maybe answer a couple of the questions. It's already 1/4 the hour. I'm going to answer a couple of questions maybe. So I'm just skimming over. I've got a sheet here. Yeah. Is there a reason why you don't use the flat bottom centrifuge tubes? Or is it that you just whatever you have? Gotta be honest with you, those centrifuge tubes, these are the only ones that I have. The wider ones you see over here, this one is also not, but they do have flat bottom ones here as well. And when I bought them from, I think over Amazon, half of them had a flat bottom and half of them were conical like this. So this was actually whatever I just got. But those I don't have flat bottom ones. I know you can chemically fix things with pure ethanol. Is it possible to do the same with isopropyl alcohol? I suppose so. Isopropyl alcohol contains less water. Is also more expensive. What alternatives are there to crystal violet? Well, in the case of gram staining, there is no other alternative to my knowledge because the crystal violet reacts with the iodine. If you just want to use anything for staining, then what I recommend to indeed take fountain pen ink and try staining. It does work. For example, cheek cells can be stained with fountain pen ink. Methylene blue is a very common one as well, maybe a little bit more readily available. So I've experimented also around with, I don't know, there are certain colors, food dyes, if you're able to get them in pure form, for example. Yeah. So for hobby reasons, there are purposes you can try with all sorts of inks as well. Yeah. And it depends really with what they react with. Yeah? But the traditional stains, the official ones like methylene blue, crystal violet, of course, they're used, of course, in a laboratory setting. Yeah. Yeah, the function of iodine is to react with the crystal violet. I mentioned that already. Okay. I would like to know what gram-positive or gram-negative actually means. I hope I explained that. What does the word gram mean in this context? It means basically simply the guy who discovered it. Mr. Gram, the Danish scientist back in the 19th century. Okay. Okay. Greetings from a veterinarian in Algeria. I really enjoy your content. Very interesting. Will this live stream be available to watch later as a replay? Yes, the live streams are always available later on. Are there categories other than gram-positive and negative? Yeah, sure. Depending on the stain that you use, or depending on a certain chemical reaction that the bacteria are able to do. For example, catalase positive or catalase negative is the presence of catalase. Yeah, so there are a whole range of different things that you can test for. Okay, so what I'm going to do now is I would like to quickly flip through the comments here, because it's. And then I slowly do want to stop, but it could be that maybe I'm skipping or overlooking some of the comments here. Okay. Yeah. And otherwise, I'm going to call it a day. Yeah. So I suspect it's a species that only lives in horses. There are several species of parasite eggs in there. I've seen the catalog or list. I mean, there are, with a reference, there are several of them. Yeah. Okay. And okay, let's have a look. Yeah. We've tested hedgehog feces for roundworm eggs. Yes. It smells like horse. Microbehunter 2026. There's a quote collections. Yeah. So I'm just quickly skimming through here, but I think perfect to watch while eating dinner. Somebody commented. Yeah. My brain isn't picking up any information at 4 a.m. I tell you, honestly, 4 a.m., I cannot believe it that some of you actually stayed up that long. Yeah. So, gram-positive, gram-negative. I think I covered pretty much everything, I hope at least. Okay. So I would like to then do the following. I hope, thank you again for having joined me. I personally always like making these streams. I think I myself learn a lot here, because it also kind of forces me to keep myself a little bit experimenting. I noticed today's with the horse manure was a little bit different. Not quite as spectacular maybe as some of the beautiful crystal pictures that we sometimes make, but still I found this quite interesting, because I actually did learn something from the student who showed it to me yesterday, and that's why I decided to kind of follow up on this a little bit. I'm going to say all the best, happy microbe hunting, as always. Hope to see you again next week, and yeah, have a nice week. Bye bye.